amino acids to the mRNA. So genetic code is essentially redundant. So you have 64 possible codons, you have 4 bases, and there's 4 cubed, makes 64, so 64 possible codons coding for 20 amino acids. It's essentially the genetic code is redundant, it repeats itself. And it's possible because the third base is actually not as strictly enforced as far as attachment goes. The third base is called a wobble base. Any questions so far? Real quick. The third base is called a wobble base. And real quick on the bases. So you have the codon, third base of the codon, you get U, C, A, or G. And you got U, C, A, or G, and anticodon, G, A, U, or C. And you know, U attaches to G, U attaches to A, and A attaches to U, G, and C. So it's also that A and U is kind of unique, two unique nucleotides in their importance. So, each specific amino acid is captured using a unit cell tRNA synthetase. It's a hard word to say. And there's 20 different synthases for the 20 different amino acids. Each one has its own unique synthetase. That's what hard to say. So what happens is the amino acid will bind to the synthetase, and then the GTP will bind to it. And then it's and then the tRNA will come, and the GTP gets phosphorylated. It gets cleaved off the GDP, and that basically is the coupling reaction that allows the covalent attachment of the polypeptide to the tRNA. It's over here. So there's a covalent attachment that's powered by glucose and GDP. Energy coupling. And it's up to synthetase is really important because that's the one that's going to ensure that the correct amino acid gets to the mRNA. Now the functional part of the whole process is the ribosome. It's the most important part. How many subunits are there to the ribosome? 2? Okay. Yes, right. That's right. Large and small subunit. Okay. And where do the subunits come together? Anybody have an idea? So you have 2 subunits, the large and small subunits, and they're actually separated in the cytoplasm. And then the mRNA will bind to the small subunit, and then the mRNA will bind to the small subunit, and then the large subunit will bind after the mRNA, the tRNA binds to that. It's actually a process of them coming together. And ribosomes are actually pretty similar to eukaryotes and prokaryotes. And basically, like evolutionarily, we're more complicated as humans than we are as bacteria. So we have 4 subunits versus the 3 subunits. And actually, for your health professionals and students, antibiotics can actually target the ribosomes and alter and stop and halt translation based on size. So if you see tetracycline, what happens is you have these, you can identify the fact that a prokaryote has 3 subunits to this 3 RNA molecules in this ribosome, and that a eukaryote has 4 RNA molecules in the ribosome. And because of that difference, it can actually bind to it and block translation. And what do you think what's going to happen when you block translation in a bacterial cell? It's going to die. It can't live anymore. Okay. Ribosome is really important. Okay. And specifically about the ribosome, the small subunit, and then you have the large subunit which has the binding sites for the tRNA. So small subunit, and the mRNA is going to bind, and the large subunit is going to bind to that. The tRNA is going to bind to that small subunit, to the mRNA, small subunit, and the large subunit is going to bind. And you have the A site, acyl site, for the acyl tRNA synthetase to bring in the protein. P is the polypeptide site, essentially making elongating the change. And E is exit site. So this is where the This is where the protein is going to leave. So, okay. Actually, so new studies being done shows that our RNA, so your ribosomes, are actually really important to catalyze the activity. So it's not the proteins that are added to the ribosome, it's the ribosome and the RNA itself that actually catalyze the reaction. So, things going on inside, so in case you guys decide to do research into this. So, building a polypeptide, what are the 3 phases of building a polypeptide? And show the answer of Bill. Anybody? Besides, I get your name? Huh? Anybody else know what the 3 parts of translation are? Yeah, correct. Good. So, divided into 3 stages. So it begins with initiation. So basically, the mRNA is brought to a small subunit. mRNA is brought to a small subunit. And what's the start codon, by the way? What is the start codon? AUG. So AUG always goes from thymine. So unique aspect about every polypeptide you can have thymine. So AUG binds, and you have an anticodon. tRNA recognizes this anticodon, so tRNA binds to the mRNA, to AUG, and with that, it goes for methionine. So methionine starts off with the P site. And when this binds, GTP has to be hydrolyzed so the binding could actually occur. So that's initiation. And initiation factors, well, then after the tRNA and mRNA are together, will actually bring the large subunits. So you have extra proteins that help in the initiation. And then you go through elongation. So you have your protein that bonds. You have your protein, or your tRNA that binds to the P site, and it has a part amino S on there. And next comes in the tRNA that binds to the A site. And then so, in order to recognize the correct codon, make sure matching occurs, GTP has to be hydrolyzed as part of the regulation of this. So once the right codon is identified, GTP gets hydrolyzed and allows the covalent attachment, allows it to attach to the large subunits. And then polypeptide is able to is added. Amino acids added to the polypeptide chain. Sorry about that. So the amino acids added to the polypeptide chain from this P to A site. So basically, this is moving this way, and the ribosome is moving to the right. So it's kind of like sliding back and forth. And when this happens, the reaction is catalyzed to form a polypeptide. So right here you have one amino acid here, the other amino acid is here, and then as a condensation, a dehydration reaction occurs, and the polypeptide becomes longer, so now you have additional chain. And then this will move over, this amino acid on the tRNA will move over, and then this will occur again. And the process of moving over is called a translocation. Okay, it's called translocation. So we talk about elongation, recognition, peptide bond formation. Okay, the P site, peptide bond, P for peptide bond, P sites. A translocation occurs, and that causes the hydrolyzation of GTP, so it can actually physically move over to the next site. And then the last site is termination. That occurs at the E site, exit site. So termination is triggered by the stop codons. What are the stop codons? Anybody? Don't worry about it. No question. UAA, UAG, UGA. You go away, you are away, you away you go. That's how we're Let's get rid of somebody. Let's get that amino acid out of there. Or get that tRNA out of there. So it's triggered by the stop codon. And stop codon doesn't actually have tRNA but probably put additional amino acid on, so it's actually just a release factor. So stop codon binds brings a release factor that comes in and essentially signals to the tRNA to stop to the ribosome to stop bringing amino acids and making polypeptide. And so then the complex assembles, large subunit goes away, tRNA goes away, mRNA goes away, and everything assembles, and you have polypeptide. Okay, so a few minutes left. And also, it's not just one at a time, it's not just one ribosome binding to mRNA. Actually, have multiple ribosomes, so you actually have multiple pieces working on it. It's like a little factory machine. Multiple guys working on the same part. And important thing, minor aspects, not minor aspects, but after you make this polypeptide, It goes through folding, which is helped for by chaperones. Chaperones are on a field trip. They come by and they watch over you, make sure you're not acting all crazy and doing what you're supposed to do. So they make sure the protein flows correctly. And then there's post-translational modification. Add sugars by constellating things so you can make the actual protein itself and the actual functional shaped protein with things on the outside. Okay, so how does the signal get to how does the polypeptide get to the site destination? It occurs because of SRP. So SRP, what does that stand for? Signal recognition part. Okay. So so you have a signal recognition part. So you have the mRNA going through the ribosome, and then you have the peptide coming off. And the first few sequences of the mRNA codons actually code for a signal peptide. And that signal peptide alerts a particular signal recognition particle to come by and bring it to where it needs to go. So for here, this protein is being made into the endoplasmic reticulum. So actually, SRP sees the signal peptide, catches it, brings this whole complex, and just moves it down to the ER, and brings it to this little hole in the ER called the translocation complex. And the polypeptide is actually synthesized, while it's being made to ribosome, it's going through this little translocation process, going straight into the endoplasmic reticulum. And on the outside, once that's done, the amino acid goes aside, and everything else assembles out and floats away. So for endoplasmic reticulum, you have SRP that brings it to the endoplasmic reticulum. Other than that, you just have the proteins are floating around the cytoplasm. You have other signals that help bring it to where it needs to be. So RNA, big thing, the central dogma. You have DNA, so RNA to protein. RNA is really the catalytic aspect of the whole process. RNA is very diverse and it actually is able to catalyze its own reactions. And it actually makes a functional shape. It actually makes a functional shape if it wants to. A few types of different RNAs: mRNA, tRNA, rRNA, small nuclear RNA, siRNA, SRP. So we can tell these RNAs are catalytic. Okay, so prokaryotes, eukaryotes, big thing evolutionarily. Eukaryotes are more complex than prokaryotes. So with time, things become more complex. And they differ in many aspects. What are a few aspects they differ in? Anybody have any idea? Aspects, how they might differ between prokaryotes and eukaryotes? True. Yeah, the process of pre-RNA is splicing into prokaryotes. They actually have everything done in the same organelle or the same space, so they don't get to separate things like the eukaryotes do. We're a little more complex. We have different departments that allow us to make different products, and then modify those products to make our specific functional need. So prokaryotes, big difference. They can translate the same gene. They can translate and transcribe the same gene simultaneously. Okay, and like I said before, like antibiotics can target different genes, prokaryotes and eukaryotes. Okay, so mutations. Point mutations change the genetic material. Mutations can often lead to things like cancer, so they're